Hello guys, this is Dr. Possibility from Excel Academy. Kindly take note that Excel Academy is offering both physical and online classes. And please make sure that you register with us so that you have a chance of winning an iPhone 7. And please kindly make sure that you subscribe to our YouTube channel. In today's class, I have a simple task which is explaining the process of fertilization. I'm pretty sure in the previous class we had to explain two important cells, which is the sperm cell and the egg, which is also known as the oocyte. And we had to discover that both the oocyte and the sperm, they are formed by the process of gametogenesis. Quite alright, we know that these cells have been formed. One is formed from the female and the other one is formed from the male. But the big question is, what's the use of these two cells? Let me give you a brief story. Many of us have seen a girlfriend and a boyfriend. These two people, they'll enjoy themselves, they'll take each other for a date, they'll send a lot of messages, and at one point they'll ask themselves to cuddle together, and eventually they're going to make love. Once they make love, if protection is not used, you discover that the female who is the girlfriend will suddenly get pregnant. And we wonder, the only thing we know about that is that the male had to release inside the girl's vagina. So, what exactly happened for the female to be pregnant? So, this is what the process of fertilization tries to explain. So, what is fertilization? So, fertilization is the process by which the sperm and the egg fuse together to form what we are calling the zygote. So the sperm cell, okay, the sperm cell and the oocyte, these two come together, join together to form what we are calling a zygote. So let's quickly look at something. I'm pretty sure some of us are trying to understand. Yes, quite alright, we are saying that it's the fusion of the male and the female sex cells, which is the sperm and the egg cell. But the big question is, where does the process of fertilization take place? So I'll explain this using a diagram, which I'm going to show you in the next slide. So in this slide, you can see all this diagram is just showing the fallopian tube. So the fallopian tube is made up of four important regions. There is this region, there is the ampulla, there is that region, and there is the other region. Quite alright, all these regions are important, but the process of fertilization normally takes place at this region, which is called the ampulla. So the ampulla is the widest part of the fallopian tube, and normally fertilization takes place on the posterior, take note of that, posterior superior side of the uterus. Posterior superior side of the uterus. So when we talk of posterior, we are simply talking about the back, and superior means top. So the back and top side of the uterus. But the whole region is known as the ampulla. So sometimes a pregnant of fertilization can take place at a wrong site, and this can cause a complication which is known as an ectopic pregnancy. An ectopic pregnancy. But we are not interested in this pregnancy, but the normal pregnancy, it takes place in the ampulla, and the specific location is the superior, the posterior superior side of the uterus. Again, one thing we need to understand about fertilization is that, quite alright, we know that there is the sperm and the egg. What we need to understand that before fertilization takes place, in what state is the sperm and in what state is the egg? So you need to understand that mostly the sperm is not capable of causing fertilization. As a result, it needs to undergo two important stages. For this sperm to be capable of causing fertilization, the sperm on its own, the way it is, okay, it cannot cause fertilization. This sperm, when it is released from the male, on its own, it cannot cause fertilization. It has to undergo two important stages for it to be able to cause fertilization. These two important stages are known as capacitation. There is capacitation, okay? There is capacitation, and the other one is known as the acrosomal reaction. So there is a capacitation and acrosomal reaction. So before a sperm cause fertilization, before the process of fertilization take place, the sperm has to undergo two important stages. The first stage is involving capacitation. So when we talk of capacitation, this is taking place in the female trunk, in the female reproductive system, in the female fallopian tube. This is where this process is taking place, and normally it takes place with a duration of 7 hours. This process, it takes about 7 hours for it to take place. Okay, now this process, you need to understand that this process of capacitation, it is involving the removal of the glycoprotein coat. So you need to understand that this head of the sperm is made up of the glycoprotein coat. It's like a coat shielding what is inside the sperm. What is inside the sperm is the one which is responsible of causing fertilization. So before that takes place, you need to understand that this glycoprotein, which is made up of glycogen and proteins, okay, it has to be removed. Okay. And it's not only the glycoprotein coat which is removed, we also have the seminoplasma proteins. These proteins from the membrane, okay, because this sperm has this membrane around. So the seminoplasma proteins and the glycoprotein, they have to be removed, okay. In short, for the pregnancy to take place, you have to remove the condom, in short, okay. So when we talk of a dick, we can talk of, let's say we have a dick somewhere there which is a penis. So when you talk of the penis, if this penis is having a condom around it, for for the process of fertilization to take place, automatically we need to make sure that that condom is not existing. So this condom, in short, it is acting as this coat of glycoproteins, okay, and then this penis is just there to show the sperm. Okay, all right, let's not go much in detail. So just know that two stages are involved: capacitation and a chromosomal reaction. Capacitation, it is involving the removal of the glycoprotein coat and the seminal plasma proteins from the what? From the sperm. And this process is normally taking place in the female tract, and it takes about 7 hours. Now, after the glycoproteins has been removed and the seminal proteins have been removed, what's supposed to be there is that the 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 sperm has now been exposed. Okay, that coat which was protecting it, it has been removed. So the next thing is that, okay, the sperm has now been removed. Okay, so when this sperm has been exposed, so when this sperm has been exposed, a reaction has to take place, and this reaction is known as what? A chromosomal reaction. So this reaction is induced by Zona proteins. These Zona proteins are proteins coming from the female, okay, from the female, which is the oocyte. So the Zona proteins, so these proteins are going to induce the process of a chromosomal reaction, and this reaction will involve the release of enzyme. There will be the release of what? Enzyme. So when capacitation take place, the Zona proteins from the female, these Zona proteins from the female, they are going to induce, they are going to start, they are going to cause the reaction called the acrosomal reaction. So how will this acrosomal reaction take place? It will take place in such a way that the sperm cell will release enzymes. It will release enzymes. It's more like the sperm has nose. Okay, I'm making it in a way you can understand. It's more like the sperm has the nose, and this nose is going to smell the Zona proteins from the oocyte, and after smelling that, it will sneeze. Okay, so the sneezing is the one which here I'm referring to as the release of enzymes. So these enzymes are acrosine and trypsin. These are just enzymes which are coming from the sperm side after the sperm notice the Zona proteins. Now, the sperm has been is now capable of causing fertilization. So we need to understand that fertilization is not a simple process. It involves 3 phases. So in the next step, I'm going to explain the phases of fertilization. So there are actually 3 phases of fertilization. There are actually 3 phases of our fertilization. The first phase is penetration of the corona radiata. Okay, so the corona radiata is part of the female oocyte. It's part of the female oocyte. You're going to see it when I show you the what, the slide of the female gamete. So the first one, it is penetration of the corona radiata. So that process we talked about, which is capacitation, whereby the removal of the glycoprotein, the removal of the seminal proteins. So that process forms the first phase, okay? It forms the first phase of fertilization, whereby the glycoproteins and the seminal vesicle proteins are being removed from the sperm. They are being removed from the sperm. Now, where does this process takes place? This process is taking place at the corona radiata, and this process is helping the sperm to penetrate through this region of the of of the of the of the oocyte. Then the next phase, it is known as the acrosomal reaction. This is where acrosomal reaction takes place. Now, this acrosomal reaction, it is explaining to us, or it's making us to understand the second phase of fertilization, which is the penetration of the what? The zona pellucida. So for this penetration of zona pellucida to take place, there is this reaction, which is the acrosomal reaction. So take this right. The first thing is penetration of the corona radiata. So for the process, for the sperm to penetrate the corona radiata, they should be what? They should be capacitation to take place, okay? And then after the corona radiata has been penetrated, the next thing is penetration of the zona pellucida. So this zona pellucida, for it to be penetrated, a chromosomal reaction has to take place. Then from there, then there will be fusion of the oocyte and the cell and the sperm cell membrane. Okay, so what am I trying to say? The first thing is penetration of the corona radiata, then there will be penetration of zona pellucida, then there will be the fusion of the oocyte and the sperm. So some of you may be wondering and sitting, I failed to understand using the notes which I've used, but can you try to explain it using the diagram? I feel it is very important for me to explain the next phase using the diagram. So let me quickly use the diagram and show you how this process takes place. So please make sure that you subscribe to our YouTube channel and make sure that you register with Excel Academy. So let me quickly show you using the Venn diagram. I was teaching mathematics, so the Venn diagram is in my head. So the first thing is binding of the protein of the sperm to the zona pellucida. There is binding of the sperm to the zona pellucida. So at this point, the zona radiata has happened. At this point, the sperm has already penetrated the zona radiata, where the process of capacitation took place. So the first stage is penetration of the zona pellucida. So here you find that the sperm is going to bind to the zona pellucida. So this is the zona pellucida. So the sperm will bind to the zona pellucida so that the reaction can take place. So when the sperm binds to the zona pellucida after penetrating this corona radiata, you're going to understand that a chromosomal reaction will take place. So the second part will be the acrosomal reaction, where the content of the acrosome are released by exocytosis. So exocytosis is the process which enables the release of the acrosome. So you need to understand that the head of the sperm has the acrosome in it. So when the membrane is removed from it, when the glycoprotein is removed from it, when the the semi-inovesical proteins are removed from it. This will enable it to release the acrosome, the content of it, and that process which enable that is the exocytosis process. Okay, now the reaction is known as the acrosomal reaction. Okay, so this is the acrosome content which has been released from the sperm's head. And then from there will be the hydrolytic enzyme release, help the sperm barrel through the zona pellucida and enable binding and fusion of the sperm with the egg. So what does this statement mean? So you need to understand that after the acrosomal reaction has taken place, there will be the release of the enzymes which I mentioned earlier on, like trypsin and the zona proteins. So those enzymes will enable the sperm and the egg to do what? To fuse. Okay, it will enable the egg and the sperm to fuse, since after that reaction the sperm will be able to penetrate through the zona pellucida. So it's very hard for a sperm to penetrate the zona pellucida. Actually, it is impossible for a sperm to penetrate through this barrier called the zona pellucida if capacitation hasn't taken place and if a chromosomal reaction has also not taken place. So the first thing is zona radiata, which is this region, has to be penetrated when the sperm undergoes capacitation. Then after that, the sperm will bind to the zona pellucida, and after the binding of the zona pellucida, the sperm will undergo the process of acrosomal reaction, where the acrosome is being released, which is this content. And after that, there will be release of the enzymes such as the zona proteins, the trypsin. Those enzymes will help the sperm to penetrate through the zona pellucida. So here the sperm has penetrated the zona pellucida. So when the sperm penetrate through the zona pellucida, it will be able to meet the nucleus of the egg. It will be able to meet the nucleus of the egg. You know, in the nucleus, that's where you're going to find the DNA. That's where you're going to find many content which are transferred from the parent to the offspring. So this sperm has penetrated by after all these stages, and now the nucleus of the sperm and that of the egg, they are going to fuse together to form the what? The zygote. So with that being said, I'm pretty sure we need to understand something. After the sperm penetrate the zona pellucida, and then there is a fusion of these two cells. After the sperm penetrates the zona pellucida, there is an issue, which is the fusion of these two cells. Now, what happens when the sperm enters the oocyte? So this is where now I'm going to discuss some few things that take place when the sperm penetrate through the oocyte. So the first thing which going to take place is the cortical and the zona reaction, and then after which are the things that I've already explained. So the next thing which is going to happen is resumption of second meiosis division. So resumption of second meiosis division. So you need to understand that the egg, which is the oocyte, was arrested at metaphase 2, of meiosis. So after the sperm penetrates and it enters the oocyte, there is an issue of what? The resumption or the starting of meiosis 2, which is starting from metaphase 2. After that, the egg cell will now undergo various metabolic reactions. So some reactions, changes will now start taking place. All right. So what are the products of fertilization? Yes, fertilization has taken place. The egg cell has fused with the sperm. These two have made what we are calling a zygote. Okay. Already we know fertilization is the process which is leading to the formation of the zygote. Now, what are the result of fertilization? So the result of fertilization are that one, there is formation of a diploid number of chromosome. Remember that the egg has what? An haploid. An egg has an haploid, and the sperm also has a what? Haploid, according to what we discussed when we were discussing the process of meiosis. So the sperm has an haploid number and the egg also has the haploid number, which is 23 plus 23, which is giving us 46. So after fertilization, there is a result which is the diploid number of chromosome, which is having 46 chromosome, including 23 pairs of chromosome. So when fertilization takes place, things tend to normalize, of which there is the formation of a diploid number of chromosome. The other thing which should be understood is that the sex of the offspring can easily be determined when fertilization has taken place. So let's say if a sperm is having a Y chromosome which it has contributed, and the egg we know it has the XX. So when this sperm mates with that one, when this sperm fuses with that, they are going to form an XY, which means that they have formed a male. In a case whereby the sperm is carrying the X and the egg, automatically we know that the egg will carry the X. So in this case, it's going to form what we are calling the female. So what we are just trying to say that after fertilization, depending on the nature of the sperm which has happened to take place, which has happened to fuse with the egg, there will be a formation of either a male or a female. So this means that after fertilization, the sex of the zygote, the sex of a child can easily be determined. It can be determined. So fertilization is the one which determines the nature of what? Of the baby. Now, yes, the diploid number has been formed, the sex has been determined, but that doesn't mean that the process of fertilization has ended there. No. What happens next? What happens next is that there will be initiation of a process which is called cleavage. Okay. So you need to understand that it's only a single cell which has been formed. Only a single cell which has been formed. A zygote having two cells which has formed together. So this process of cleavage will be initiated. So when you talk of this process of cleavage, this is where now you're going to understand further what happens to that zygote which has been formed? Automatically, it's going to undergo some mitotic divisions, and this mitotic division will lead to it forming the morula, the blastocyst, and other stuffs which are going to be explained well. Okay, I'm pretty sure, and I'm 100% sure that all this explanation, some of you would need more of the explanation on the penetration of the cell when we talk of the zona pellucida. So I have my friend there who is trying to ask me on if I can explain more on the penetration of the zona pellucida. So what are we supposed to know about the zona pellucida? I'm pretty sure from the previous diagram you would see I explained about the corona radiata where capacitation has to take place for the cell to penetrate through it, and this capacitation will involve the sperm removing the what, the membrane around it. Okay, it has to be removed. The membrane around it, that membrane which is having the glycoproteins, that cord has to be removed. The cord which is on top here, it has to be removed, that cord. Okay, so I explained that. So there will be penetration of the corona radiata. Make sure that you know this diagram. Make sure that you know this diagram. Then from there will be an issue of penetrating the zona pellucida, and that will make the sperm to reach the what we are calling the what we are calling the perivitelline space, and after reaching, this is the perivitelline space, and after that it's going to reach the vitelline membrane, and this vitelline membrane, it is found on the egg cell. Okay, anyway, I'm trying to explain to you the penetration of the sperm through the zona pellucida. So we need to understand one thing. What is this zona pellucida? This boundary here in yellow, this boundary here, what is it made of? What is its function? Okay, so we need to understand that the zona pellucida, just like a sperm, it is made up of the glycoprotein shell. The sperm, we said, it is not capable of causing the process of fertilization because of the fact that the sperm has what we are calling the glycoprotein coat, which is covering the acrosomes. So now even the zona pellucida has the glycoprotein shell. This glycoprotein shell, it is protecting the cell, it's protecting the egg. Okay, it surrounds the egg. As you can see, this is the egg inside, this is the egg inside. Okay, so this egg is having the zona pellucida. So this zona pellucida is preventing, or it's protecting the egg. Actually, once one sperm fertilize the egg, these other sperms will not be able to enter and fertilize this egg more. Why? This zona pellucida, it will enclose and it will make sure that no other protein is entering is penetrating it. Okay, so what is the major function of the zona pellucida? One, it facilitates the binding of the sperm. It facilitates. Without it, they can be no maintenance. Okay, anything can enter, any sperm can enter. Okay, now that simply means that this same zona pellucida, it maintains the binding of the sperm. It maintains. One, it facilitates. How is it facilitates? It's making sure that for the sperm to penetrate through it, it has undergone the process of capacitation. This sperm, for it to penetrate through it, it has to wait for the zona protein to initiate it to produce other proteins, and it should be able to undergo the process of acrosomal reaction. It has to release the content it's having. It has to release the content it's simply carrying. Okay, so that's what the zona pellucida does. And then this same zona pellucida, it induces the process of acrosomal reaction. I'm pretty sure I'm just explaining the same things over and over and over and over. So that means that it will be very easy for you to understand this concept. Acrosomal reaction, it is the zona pellucida which is inducing the what? The acrosomal reaction. So for the acrosomal reaction to take place, this zona pellucida has to uh, has to help. Now, the question we need to ask ourselves is, what's causing? You know, I really love and I really enjoy anatomy, specifically embryology, because it tells me how I was created, how I was formed. So, what's really helping this process of acrosomal reaction to take place? Yes, zona pellucida is inducing it. But what's really causing this acrosomal reaction to take place? By the way, make sure that you take note of these functions of the zona pellucida. So let me quickly look at one of the important contents that we need to look at. So the acrosomal reaction and the binding, okay? Remember, you know, like I told you, I'm just rotating on the same thing. So remember what I said. I said that capacitation has taken place. The 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 glycoprotein cord has been removed from the sperm. The seminal-vescoproteins have been removed from the membrane. Now, the zona pellucida will release the zona what? The zona proteins. So, this process of acrosomal reaction is mediated by what we are calling the what? The ligand, of which the type of this it is ZP3, and also the protein. Now, after that, I love this. Where is this coming from? Automatically, these things are coming from the zona pellucida, the region of the egg. That's where these guys are coming from. But what about the sperm? So the sperm will release the acrosomal enzymes. The sperm will release the acrosomal enzymes. An example is acrosin. The other enzymes that we talked of, it was trypsin, acrosin. There is also trypsin, isn't it? Trypsin, acrosin, trypsin. So acrosin and trypsin will be released by the sperm. Then ligand ZP3 and the zona proteins will be released from the female. So here is the thing. The sperm is releasing the trypsin, okay? It's releasing trypsin, and then it is also releasing acrosin, okay? Then you have this side the egg surrounded by the zona pellucida. For it, it's going to release what we are calling the zona protein, which is coming from the zona pellucida. So you have the zona proteins and you have the ligands, the ZP3, ZP3. Okay, Zambian police. Sorry, that's not what it stands for. So look at that. These two have caused that. Now, after that, this sperm now, it's wanting to penetrate. This sperm here now, after it passes through this zona pellucida here, it now has to penetrate and touches the membrane. What's going to happen here? It has touched the membrane. Okay? It has touched the membrane. After touching the membrane, what's going to happen? Look at that tail. This tail will remain. This tail will surely remain. We'll talk about that later. So, the sperm has penetrated the zona pellucida. The sperm has penetrated the zona pellucida. After it penetrates through the zona pellucida, it's going to be in contact with the plasma membrane of the egg, which is the oocyte. Now, the permeability of the zona pellucida, it will be under control. This zona pellucida will say, no more sperms. This sperm which has entered that soul, like most of faith for women, they'll, once they have one boyfriend, any other man who come in their life, they'll say, no, I have a boyfriend. This is the same thing which happens to the zona pellucida. Okay? Zona pellucida, once this sperm attaches the membrane, of the egg, this zona pellucida will be impermeable. It will say no to other sperms. No more sperm to enter. This sperm which has entered is simply enough with me. So, the permeability of the zona pellucida, it changes. Look, what did we say? The zona pellucida facilitates the binding of the sperm. The zona pellucida maintains the binding of the sperm. So at this point, the first thing, the sperm had to penetrate through the zona radiata, and it was helped by capacitation. After that, there was an issue of the sperm to penetrate through the zona pellucida. So when it attached the zona pellucida, this sperm had to be helped by the ligand ZP3, the zona proteins, and these other proteins from the male. Okay, so from there, the sperm will penetrate through the zona pellucida. Once it penetrates through the zona pellucida, it will do what? It will be in contact with the membrane. By the way, zona ligand ZP3 is an example of the zona protein. Ligand ZP3 is an example of the what? The zona protein. There are a lot of zona proteins out there. So the zona pellucida has become impermeable after the head attaches. That means that the tail will remain outside. So what next are we supposed to pick from there? So once the sperm has attached itself to the membrane of the oocyte, you need to understand that this contact, it enables the release of the lysosomal enzyme from cortical granules lining the what? The plasma membrane. So this plasma membrane, it is having lysosomal enzymes, okay? It is having lysosomal enzymes. So the lysosomal enzymes will now be released. I'm sure these are the lysosomal enzymes. So these enzymes, they are now going to be released. So these granules here, they are going to release the lysosomal enzymes, okay? So now, after the lysosomal enzymes are released, in short, what are we supposed to understand? We need to understand that what is the function of these enzymes, these lysosomal enzymes? We are saying the lysosomal enzymes will be released, okay? The lysosomal will be released after the sperm is in contact with the membrane, okay? And these lysosomal enzymes are produced from the cortical granules. These granules here, they are going to produce what? The lysosomal enzymes. So the question is, what's the function of these lysosomal enzymes? So these lysosomal enzymes, they're going to change the nature of the what? Of the zona pellucida. In the previous slide, I gave a statement where I said, once the sperm binds to the membrane of the oocyte, the zona pellucida, it becomes impermeable. The zona pellucida, it becomes impermeable. So what's causing this zona pellucida to be impermeable is these lysosomal enzymes which have been released from the cortical, okay, granules. These granules, they are going to release the lysosomal enzymes. So once these lysosomal enzymes are produced, this zona pellucida will become impermeable. Okay, so this impermeability will prevent more sperms from what? From entering. This zona pellucida, when it becomes impermeable, it means that there will be no entrance of other sperms. The oocyte has said enough. Enough. That's why when a girl has a boyfriend, she'll be very, very proud and pride. You should develop that pride and stubbornness, whereby when another man comes, she'll say, no, no, I have a boyfriend. It is not that they do that behavior as human being. No, even their oocyte, even their egg cells, that's how they behave. Once the sperm attaches there, this boyfriend, the sperm attaches there, it will release the pride, and that pride is what we are saying, the lysosomal enzyme. This lysosomal enzyme will prevent more sperms, will prevent more boyfriend. She'll prevent more boyfriend from entering or being part of her life. So when the examination is done, when the examination was done, it was found that some of the sperms were embedded in the zona pellucida, and only one, who is you, who is me. I came from only one sperm. You came from only one sperm. Your friend came from only one sperm. No one came from two sperms. The rest, actually over 300 million of sperms are produced, and those 300 million, between 200 and 300 million are produced just by a single shot in a 3.5 mil of sperm are there. But only one sperm is what? is capable of causing fertilization and form the child when they fuse. All right, I'm pretty sure, guys, we have discussed a lot about the process of fertilization. We talked about the sperm, how incapable it is, how it's not able to cause fertilization. We said it has to undergo 2 steps. We had to say it has to undergo capacitation and it has to undergo a chromosomal reaction. Then from there, we talked of how the sperm penetrates through the zona pellucida, through the zona radiata until it reaches the oocyte. And once it reaches the oocyte, you need to understand that the process of fertilization, the process of fertilization will take place. So you need to understand that what simply fuse, it's not really the oospem. Instead, there is the nuclei of the oocyte and the nuclei of the nuclei of the sperm. So the nuclei of the oocyte and the nuclei of the sperm. So it is the sperm nuclei. Maybe let me write it down. The sperm nuclei and the oocyte nuclei. Okay, these 2 nuclei are the ones which fuse to form a zygote after all these processes have taken place. So please, guys, make sure that you subscribe to our YouTube channel and make sure that you contact me on that number on WhatsApp, and you can also follow us on Facebook. And please make sure that you register with Excel Academy. Thank you very much. This was Dr. Possibility from Excel Academy. Have a blessed day.